My name is Sara Linker. I'm a postdoc, as Philip said, at the Salk Institute in the lab of Rusty Gage. And the Salk Institute is a really nice place to work. It's in La Jolla, and it's a place where a lot of neuroscience research is being done from a lot of different angles. So people study genetics and induced pluripotent stem cells and evolution at the same time. And there's a lot of collaboration, and it's really a good place to do neuroscience research, which is really what the focus of my talk is going to be today. The title of my talk is Studying Primate Evolution in a Dish, an Exploration into Neuronal Diversity. So to give you a little background on what I'm going to talk about, I'm going to talk about neuroscience and gene expression, and basically how, when a neuron is first being formed in the developing embryo, how gene expression patterns are beginning to be defining the neuronal subtype, or the type of neuron that's being developed. And specifically, I'm going to ask this question with respect to looking at different species. So we're going to take neurons from humans and neurons from gorillas and rhesus macaque, and we're going to compare how these different neurons progress through differentiation of a neuron and see what different aspects are present due to the species that we're looking at. I'm going to tell you that for the most part, there's a lot of conservation. So as you may expect, as you transfer from an early progenitor cell to a final mature functioning neuron, there are a lot of conserved gene expression profiles that are exhibited. And this is because a neuron is a highly specialized cell, and so you need a lot of highly specialized function. For example, synapses, or the ability to transfer chemical information between two cells. That's going to be present no matter what species you're in. But independent of all those conserved gene expression profiles that are present in the neurons, we can actually pick up expression profiles that are specific to the neuron specific to the species that neuron is coming from. So I'm going to show you some of those profiles and further tell you that human neurons have a very specific sort of phenotype associated with the neuron, and that it maintains this sort of neotenic phenotype compared to the other primates, meaning that it stays younger, or it looks like it might be staying younger longer.
Everybody hearing well? Okay.
Thanks. So one question we have to start off by asking is, why are we even studying primate neurogenesis? So this comes back to that big question of what makes humans different, what makes us what we think of as unique. And we often talk about this principle, or this thought, that we have this higher complex, potentially social structure, more highly complex thoughts. And I sort of wanted to illustrate that with this photo here. So this is a picture of the National Library in Vienna. And what I like about this is that you don't see a single real live human in this picture. But I don't think anyone here would mistake the aspects of this picture to be created by any organism other than a human. You see history, you see accumulation of information over the ages, you see creativity, you see what is information that's required to be passed down from generation to generation in order to build scientific principles to allow for electricity and architecture and sculpture. So what is the basis of the human being that allows us to have these highly complex, to build these highly complex structures? And a lot of people bring this back to the brain and identify the brain as being one of the major areas that allows us to be different. Another reason to look at the variation between primates is a little bit more directed of a reason. And this is that if we understand the variation in neurogenesis or neurons between different primates, that may help us to understand the variation between individuals. For example, individuals that may have a neurodevelopmental disease. Right now in the medical genetics world, it's really hard to pin down what genes are really responsible for the phenotypes that we're seeing. But if we look at these really broad areas of variation, we may be able to get clues as to what's going on at these lower levels of variation. So one of the biggest differences for humans with respect to brains is that we have a much larger brain. So this, let's see if I can use nope. Is there a pointer? Okay, so on the top left, you can see a brain of an adult rhesus macaque, which is about the same size as the brain on the right, which is the brain from a developing human being. As that developing human, human develops, we move down to the bottom, and you see that that brain expands, and we get more gyrification occurring in the brain. So it may not come as a total surprise to you that the human brain is larger than the brain of a rhesus macaque. A rhesus macaque is a much smaller animal than a human, but we also have a larger relative brain mass compared with respect to our size. So on the x-axis here, we have the body weight of a number of different organisms, and on the y-axis, you have the brain weight of that organism. And you can see that there's a relative linear scaling of brain weight and body weight. So usually you can predict the brain weight of an organism just given its body weight. But if you were to do that for the human, you'd be wrong, because the human it is along that blue line. Do you have a pointer by any chance?
I just realized that
Okay, which is above the line. So if you were to predict the brain weight of the human, you'd actually be under-predicting compared to what we really have. So the human brain is larger compared to its relative body mass, and we also have more surface area than other species. So here I'm showing a section of a human brain, a rhesus macaque, and as a mouse. And on the bottom, you can see the mouse doesn't have any gyrification patterns. As we move up towards the rhesus, you start to get a little bit more folding. But if we look back at our original image on the left, you can see that even the folding that you see there in the rhesus is only akin to pretty much prenatal human, and nowhere near the amount of gyrification that we have in the adult human brain, which you can see on the top left. So the brain size has increased, we have increased surface area. Well, what about subregions that are specialized for functions that we mentioned earlier may be involved in complex thought and complex processing? Well, one of these regions is the neocortex. And it's thought that the neocortex is also expanded in upper order primates, including humans. So this is a figure taken from Barton and Harvey in Nature 2000. On the x-axis, they have the brain volume. So before we saw body volume, this is now the brain volume. And on the y-axis, we have neocortical volume. And you can, so the open triangles are insectivore mammals. The closed circles are Strepsirini primates. And the open circles are Haplorini primates, which we are included in that group. And you can see, as we saw before, there's a relatively linear increase of neocortical volume when you increase the total brain volume. But there's an interesting shift, an increase in the intercept of that value as you go increase in order through the primate branch. So this means that something has occurred in evolution, something at the genetic level has occurred that has allowed the neocortex to expand even more than our whole brain has been allowed to expand. So we have an increase in brain size, but also increase in these specific regions. So what are the reasons for the increase in brain size? How do we get at what is actually the cause? Well, I'm sure you'll hear in other lectures some reasons. There are a lot of hypotheses out there evolutionarily to suggest why we may have larger brains. I'm only going to focus on two very small topics today. One is protracted development, or neoteny, and this is the thought that humans are just allowed to develop for a little bit longer than other primates. And that allowance to develop our brain longer just allows us to expand a greater volume of the brain. Another reason is the expansion of the regions of the brain that proliferate. The regions that are going to add more cells to the human brain have expanded in upper order primates, and so this may be also providing a reason for why we have a larger brain. So to talk a little bit about neoteny, or protracted development, I put a little image up there of a chimp and a human, and they were raised at the same time, at the same age, and they're sort of growing up together. I just like this image because it sort of looks like the chimp is revealing the larger brain of the human. But the idea here is that as the human develops, our brain develops larger and slower than other primates. So you can see on the right, we have a figure showing age in years. and the size of a human brain compared to the size of the pan troglodytes or the chimpanzee brain. And you can see that the mass increases largely during early development and through early postnatal development. Yeah. Yeah, no, I tried. It doesn't show up on the screen.
Oh, it doesn't? Oh,
no.
Because of the white background.
Sorry. No, it's okay. So we have protracted development seen on a scale of weighing the brain, but there's some evidence by Samal et al. In 2009 where they actually went down to the molecular layer, or the molecular aspect, and asked how genes are changing through development, and whether or not they could find some genes that sort of were attributing to this slower growth in humans. And so the first thing they did was they took tissue from human postmortem brain and chimp postmortem brain over multiple years, so that x-axis ranges from 0 to 40 years old, and the y-axis is the change in postnatal gene expression. So the higher slope here on the left means that you have a lot of genes that are changing really fast. And as you taper off in older age, it means not much is changing, and so you're relatively staying stable. And what they did is they found a few genes that actually exhibit a slower growth in humans compared to chimp and rhesus, this was indicating to them that maybe there was this neoteny. Maybe it wasn't just a phenotype at the tissue level, but that there was something at the molecular level that was delayed in human progression compared to other chimps. The only problem here in this study is that they're using a really large range. They're going from 0 to 44 years. And yes, they found genes that are slower in humans, but they also found genes that were quicker in humans. So they found evidence of a lot of different dynamics occurring. And the growth in the size of the brain is really more towards the earlier part of development, not so much at these later ranges. So what we need to do is we need to focus in on the time point in development where neurogenesis is occurring. We need to focus in on this time when proliferation is occurring and when the brain is really growing. So what does this time point look like? Bless you. So on the left, we have a tissue section of a rhesus macaque's brain. It looks a little different in development than it does in the mature brain, but you can trust that this is the somatosensory cortex, and you can start to see a little bit of a layering pattern in that cortex that will eventually set up layering pattern for the adult mature cortex later on. There's a little zoom-in view where you can see many different layers, and these are all the proliferative layers. They only exist early in development, and then they morph into the mature layers later on. But these layers are the ventricular zone, the subventricular zone, the subplate, and the cortical plate. Those are the 4 layers you're seeing. And the interesting thing here is that this subventricular zone, those 2 lower regions there, they're split into inner and outer, and that is only present in upper order animals, in animals where you start to see the gyrification pattern. And people think that this region, these 2 regions that have split, are starting to build more cells and enable for this expansion of the brain. And this is shown here as a little comparison, where you can see the monkey, the rhesus macaque brain compared to the mouse. And you see that early in development, they have pretty much the same layering pattern. The different colors are different layers of the brain. And as you move to the right, you move more towards differentiation and development, and you start to see a really quick expansion of this outer subventricular zone in the primate, but not in the mouse. And so this is why people think that this is a large part of why the brain has expanded. So what is going on here? What are these cells? What are they doing? So this is where we finally get to neurogenesis. So on the left, we have the neural progenitor population, and those are the cells that make up that proliferating outer subventricular zone region. They're stem cells of sorts. They're multiplying and creating the cells that will eventually become the neurons. So in the first step here in early development, they're proliferating, and then they start to migrate up through those layers that you saw the picture of. And on the final right side, they differentiate into those neurons that we know and love, that make synapses, that make the computation that allows for higher order processing. Now this slide is pretty important for the rest of my talk to understand this, so I'm just going to repeat it one more time so we're all clear. There are two major cell types that we're concentrating on for this talk. They're the neural progenitor cells that are proliferating, those stem cell-like cells, and then the neurons, which neurons don't divide, right? They just stay and they perform a specialized function. So far I've told you about the expansion of the human brain, that we have a larger mass, a larger relative mass, a larger surface area, and that some possible reasons for this could be the prolonged period of neurogenesis, an increased number and type of neural progenitor cells, and that we would like to now examine the differences between primates. Now, some people have done this, as I've shown before, and have enabled us to trace some of the molecular underpinnings of early neurodevelopment and brain size, but we really want to be able to look at this at a deeper level. And historically, this has been sort of difficult to do. One reason is that you have to be able to acquire postmortem samples from prenatal brains of multiple different species. And also, a lot of these studies are biased by environmental factors. So for example, different species may not eat the same diet, they may not experience the same environment while they're being developed. And so we want to be able to erase all of these confounding factors, keep everything steady, and just ask the question of how is the DNA, the evolved genomic code, affecting the phenotype of this cell and how it's growing. So luckily, we have a tool available to us in the past decade or so, and the lab that I work in is very specialized in this technique, and that's the technique of creating induced pluripotent stem cells. So these are stem cells that are actually created from a differentiated cell. So you take a piece of your skin, a skin biopsy, and you put that skin biopsy into a dish, and you add just a few transcription factors to that dish, and what happens is those transcription factors get into the chromatin, and they remodel it and rewind developmental time until that skin cell or that keratinocyte has gone back to become an induced pluripotent stem cell. And since we induced this, we call it induced pluripotent stem cells. You can then take these pluripotent stem cells and make them into any cell type, or mostly any cell type of the body that you want. You can turn them into neurons, or heart cells, or pancreatic islet cells. And people use this now a lot for disease studies and modeling. And it's what we're going to be using in what I'm going to tell you about today. So as I mentioned, many people have been using this since its invention. On the left are cardiomyocytes. So they took induced pluripotent stem cells and created heart cells that are spontaneously beating together, contracting together in the dish. So these have been generated from skin cells that then became iPS and then were converted to heart cells or cardiomyocytes. On the right, you can see induced pluripotent stem cells that were then created into neurons or differentiated into neurons in order to study the difference in individuals who are diagnosed with schizophrenia. So on the left column, you have the induced pluripotent stem cell. In the middle is that neural progenitor cell, that proliferating population I talked about. And on the far right is the neuron. On the top are controls, and on the bottom are stem cells created from individuals who've been diagnosed with schizophrenia. And the reason why I'm putting this one up, this figure up, is because I really think it shows that we don't know what causes schizophrenia. We have some ideas of some genes that are involved, but we know that there's a genetic component and an environmental component. And what Kristen Brennan was able to do here was remove all of the environmental component that's associated with developing schizophrenia and only take that genetic component, even though we don't know what it is, but isolate it in a dish. And she was able to discover different phenotypes associated with neurons derived from an individual with schizophrenia and an individual who had no previous documented illness. And so we're trying to use this same principle to understand the differences between primates. So Carol Marchetto in our lab developed iPS cells for chimpanzee on the top, bonobo in the middle, and human, and identified that even at this early iPS level, well, one, that she was able to make these cells. So this was really a breakthrough, and she published it in Nature in 2013. And they found that there were thousands of genes that were different, even at this really early stem cell state, between the chimp, bonobo, and human. And the interesting thing is that they're not just all different from each other, but if you look on the right, these are, it's a heat map of gene expression, where each tile is a gene, and the more red it is, the more highly expressed, the more green, the lowly expressed. But you can see that chimp and bonobo look similar to each other, as we'd expect, because they're relatively close to each other in evolutionary time, and they're very far away from human. So it's not just that there's some random pattern accumulating in these iPS cells, it's specific to the species of origin. Recently, she's collaborated with the Otani group, and they came out with this paper this year in Cell, where they took those iPS cells and they converted them into neurons. So as we move down, we have iPS at the top, some intermediate populations, the neural progenitor cells that I told you about, and then neurons. And they looked at these in human, chimp, and rhesus. And they then took the physiological recordings of these neurons. So they stuck a little pipette into the neurons and asked how they're spiking. And that's what we see here. Hopefully you can see there. If you can't see it, the box sort of circles where spiking is starting to happen. What they did is they differentiated the neurons for months at a time. And they found that rhesus neurons started to spike earlier at about day 40, compared to the chimp, which started to spike at about day 50, and human at about day 60. Sort of starting to indicate that at the molecular level in a dish, we can already start to see this neoteny that we're talking about, where humans have a little bit of a delay compared to the other non-human primates. So now it comes to the question that we want to ask, or I want to ask, which is what are the molecular events, or what at the DNA level is causing these differences? So before we get there, I just wanted to throw up a plot. We talk a lot about differences between humans and chimps and how we're so similar. This is an exact plot of that. So the x-axis is the entire human genome at a base pair level. And as you move across the genome, you see how different it is from either chimp, gorilla, or orangutan. And there's really only about a 1% difference at any location from the chimp. This changes depending on whether or not you're in a gene. A gene is going to be more highly conserved, but on average, you have a pretty high conservation between human and chimp. But there are many regulatory regions that have been modified. So perhaps not the code of the gene itself, but the regulation of when that code is expressed is really what we need to track down. So in order to do that, we're going to look at gene expression in the neurons of these primates as they're developing. So Carol Marchetto, again in our lab, developed iPS cells for human, chimpanzee, bonobo, gorilla, and rhesus. And this sort of separates out nicely across primate evolution, with human and chimp split being around 5 million years. If we split to gorilla, it's about 8 million, and rhesus macaque, we have about a 25 million year split. So we have a pretty nice spread across primate evolution. And she took these cells and differentiated them into embroid bodies, an intermediate neural rosette population, and then finally those neural progenitor cells, those cells that are fastly proliferating and dividing. So we do RNA-seq here, which allows us to look at gene expression, and that's when we start to look at our data and see what it means. So this is where I come in. I'm more of a computational biologist. I work at the computer, so I get to look through all this data and discover what it all means. And one of the first places that we need to start is making sure that we're really figuring out what the differences are at the expression level between a human and a non-human primate, as opposed to some artifact due to the way we're doing the experiment. And this is really important with respect to looking at gene expression, because what we're doing is we're taking little pieces of that gene and we're mapping them back to some reference genome that has been made for the species. So for example, you take a gene from a human that's expressing in a human and you map it back to the human reference genome. Well, that's all and good. You expect to get a pretty good mapping when you do that. But what happens when you take a RNA from a chimp genome and you map it back to the human? Well, when you do that, you may not expect a perfect mapping, right? So you may lose some information, and we have to make sure that that loss of information doesn't make us think that we have less expression of that gene. So in order to do that, I sort of spread this error across all of the species, and we map our RNA to every species that we have available. So in green, we have the human genome. In orange, we have the chimp, then the gorilla in purple, and the rhesus in blue. So we map all of our species RNA profiles to all of these genomes and filter out only the reads that are consistently mapping across all species once. And this allows us to know that we are only looking at regions of the genome that are conserved and at least similar between all of the species. We then compare gene expression profiles between mappings to make sure that we don't have any remaining bias. We get rid of any remaining bias we have, and that's shown here. So everything I've said, it could be a little bit confusing, so I put up a explanatory figure to go over it. So I don't know if anyone here has ever looked at at a browser view of a gene, but what we're seeing here in blue is an exon of a gene, and the blue line in between is the intron. And when you look at gene expression, you'd expect to only see exons, right? Because the introns are spliced out and they're gotten rid of. So what we see piled up over those dark blue boxes, that's the actual RNA-seq information that we're seeing aligned to the genome. On the top, we have a gene VASP that we see is only aligned to the human genome in that top row. As we start to align to further primates, if we include chimp or gorilla or rhesus, that gene completely goes away. And so this means that this is not a gene that's correctly aligning across the species and will give us some error, so we don't look at that information. Conversely, if we have this other gene, VAMP2, that aligns well to every species no matter what we do to it, that means that we trust when we see lower expression that that's really lower expression, and it's not due to the alignment bias of the gene. So we'll use that one. Now, something may come up when you think about this, which is, well, we want to see differences between primates, but we're getting rid of the genes that are different between primates. So what does that mean? Well, we have to do this because we have to be able to compare across species, but I did take a look at what genes we're removing by filtering out this way, and we actually keep about 94% of our genes. So most of our genes we're analyzing, it's fine, and we're getting rid of just some ribosomal genes, some pseudogenes, genes that are regulating the function of other genes, which we should be able to pick up. So now that I know we have a good set of information, we compare the species. So this is a correlation plot of that neural progenitor cell for all of the species that we analyzed. On the top right, you can see in green are the humans. Next to that are the gorilla, then the chimp and bonobo, and then finally the rhesus in blue. The more yellow a box is, that means that the samples are more highly correlated with another. And you can see that the humans are highly correlated with another, as you would expect, between the species. And actually, it's true of all of the species. They're highly correlated within one another. But interestingly, gorilla is actually sitting closer to human than the chimp or bonobo, which is not what we see when we look at the whole transcriptome. And this is really reproduced in many ways throughout the analysis. Sometimes we see gorilla is sitting closer to human. Interestingly, sometimes we see that rhesus macaque and human have a more similar expression profile than they do to gorilla and chimp and bonobo. So it really depends on what genes you're looking at. But overall, it tends to look like humans sit closer to the chimps, the bonobo and the chimps, than they do to gorilla and rhesus. And this can be seen in this gene here, GATS, where you can see a clear delineation of evolutionary time by gene expression. So we have higher expression in the human, followed by the bonobo and the chimp, then the gorilla, and finally the rhesus. And just to drive home that this is not an alignment error, because that is always a worry, I wanted to show you guys that no matter what species we align this to, we get the same alignment. So this is true difference in gene expression that's associated to evolutionary time. So now that we know what the gene expression looks like and we know the correlations, we want to ask what kinds of functions do these genes have? And we do that by looking at the literature and asking if there are known groups of genes that come up. And there are many. As you might imagine, there are thousands of genes that are different between the species. But one group of genes really stuck out to me. And this is a group of genes that's involved in patterning the early brain. So this is an image of the early brain on the right. On the bottom is the more hindbrain region, and on the top is the more forebrain region. the genes on the left are patterning that brain. So if you have, for example, GBX2 expression and EN1 expression, you expect to have a more hindbrain phenotype. And if you have a more 6-3 and OTX2 expression, you get a more forebrain phenotype. And interestingly, a lot of these genes were coming up in our differential expression analysis, where from left to right, we have human to rhesus, and from up to down, we have more forebrain and more hindbrain genes. And you can see the genes that are higher in human tend to have a more hindbrain phenotype compared to those in rhesus, which have a more forebrain phenotype. And this is one indicative of differential regulation already at this MPC state, or neural progenitor state, but also that perhaps this is already a sign of neoteny, and that these earlier brain regions are present in human, while the brain regions that develop a little later in time are already present in the rhesus. So since we saw this, we wanted to ask whether or not this is maintained throughout differentiation of that neuron. So again, Carol took iPS cells, differentiated them to embroid bodies, all the way to neural progenitor cells, and now neurons. So now we're going to get our functional cell. And she differentiated them over 2 months and collected RNA every 2 weeks. So we have 2, 4, 6, and 8 weeks of neurons, tons of information. And one way to plot this is with principal components analysis, which is basically just takes all of your data and finds the pieces of the data that make the biggest difference and gives you a score based on that. And so we plot each score, the first 2 scores versus each other, and that tells us where the most differences are coming from. So on PC1, we can see that the biggest difference is between cell state. So on the left, all of those circles are all our neural progenitor cells, and on the right are all of our neurons. And this comes back to what I told you at the very beginning, that independent of what species you're from, you are derived from, a neural progenitor cell is a neural progenitor cell, and a neuron is a neuron. This is a highly conserved state. As we move up on principal component 2, we see a separation of the species. So the second most important piece of variation is the species. On the bottom in green, we have human, and they sit next to the bonobo and rhesus in red and pink, then the gorilla in purple, and finally, far away is the rhesus. So not only do they separate on species, but they actually separate based on evolutionary time. And I know I have convinced you already that that is not a methodological error. This is not due to alignment. This is gene expression differences that are due to evolutionary time. So first, to understand what's going on here, I took a look at what differences are underlying this conserved transition from neural progenitor cell to neuron. And these are the genes marked D for differentiation. And on the right is a heat map of all of these genes. So each column is a gene. Each row is a species. The top rows are neural progenitor cells, and the bottom rows are neurons. In red is higher expression, and blue is lower expression. So you can see that independent of what species you're coming from, the neural progenitor cells of that species have higher expression of a certain set of genes, and when they become a neuron, they have higher expression of that set. So this is independent of species. And if we look at what kinds of genes they are, these are genes that are expressed in the cell cycle, differentiation processes, the things that we would expect a proliferating cell to be involved in. So what I'm showing here, each dot is a gene, and they're sort of clustered based on their proximity and function. And on the right are our genes for neurons, and they tend to cluster in genes involved in making a synapse and making a mature neuron. So they're doing exactly what we expect to do. All of the cells are turning from an MPC to a neuron. So the next thing we want to do is identify whether or not we are in a place in developmental time that is interesting to us. So remember before I showed you that plot of neoteny where we looked at from 0 to 40 years, and I said that that might be a little bit long? Well, we want to know how old our neurons are in the dish. We don't really know what that's related to in real developmental time. So I created a classifier to predict what point in developmental time these cells were at. And we did that by taking post-mortem samples, publicly available data of post-mortem samples from a variety of ages, and plotted them again on this principal components kind of plot that separates out major components of variation. And the major component is time. So this first x-axis here is really separated out very far. All of the samples are very far from one another, and that's associated with the post-conception weeks, or how many weeks old that individual is from conception. And we can see that the further we go to the right, the older that individual is. And so the genes that make this plot happen can be used to predict how old our samples are. So two of the genes that came up in this analysis are shown here. Again, blue is low, red is high, and to the left are younger samples, and to the right are older samples. And you can see that for BST2, this is from postmortem data, it increases in a real human being, and ChRNA5 decreases through age in a real human being. In our dish, these same genes are going up and going down. So BST2 is going up, and ChRNA5 is going down. So this means that we can use this information to predict where in time our cells are. And we can, it's not just these two genes we're using a lot more genes, about 100, to calculate this. And this is what I'm showing on the right. So each spot here is a sample from a different species. 0 are our neural progenitor cells, that first population I talked about. And then 2, 4, 6, and 8 are the neurons that we've differentiated. On the left is the predicted age that that cell would be if it was taken from a real human being. Our neural progenitor cells are estimated to be about 15 weeks post-conception. And with increasing differentiation, we get increasing age, but all of these cells are prenatal, or expected to be prenatal in mid-gestation in time. So this is exactly the time point that we want to be looking at. Another thing that we're able to do is not only look at time, but look at region. So remember I told you that those genes look like they're setting up a different region between the species? Well, we want to know if that's a pattern that's true overall. So I used the same data set to predict what region of the brain we're in. So this is an example screenshot I took from the publicly available database of a gene, HER6. And each little tile here is a tissue. And the red are regions of the developing brain that are proliferating. These are the subventricular zone and ventricular zone regions. I take all of that expression information, I find which regions are expressed to the highest. So as you can see by eye here, the SZ and VZ, and then I make a picture of the brain. And I color it in based on the level of expression of that gene in that region of the brain. So here you can see by eye, SC, VZ should be expressed, and they are in that developing brain. Now, this is sort of trivial if we're looking at a single gene. You can just go to the website, look by eye, and you don't really need to do much else. But what I was able to do is use this same approach and combine multiple genes and see where they accumulate in the brain using this data set. So if we look back at that gene expression profile that I showed you before with neural progenitor cells and neurons, we see that the neural progenitor cells tend to express genes that are in the highly proliferative regions of the brain. So this is exactly what we'd expect. And the genes that are high in neurons are in the highly differentiated regions of the brain. So they're no longer in the SC-VZ area, they're in the telencephalon, they're in the hindbrain, they're in the developing, maturing region of the brain. So now we have some tools to predict time and to predict space computationally. So the next thing we want to do is look at what is the effect of the species. So here I'm showing the principal component 2 in one dimension. It's just a little bit clearer that way. So on the left, we have in green all of our human samples, and then our chimp, our bonobo, our gorilla, and our rhesus. And this is not me imparting any kind of hopeful wishfulness on the samples. This is how they line up due to their gene expression profiles. If we then take the genes and order them based on how much they give a weight, how much they are required to make that pattern, we see that those developmental genes that I was talking about before are some of the most highly ranked genes. So I went in again to ask what genes were differentially expressed. And you're going to have to take my word for it here. But the genes on top that you see are highly expressed in rhesus in the MPC state, and then in rhesus, gorilla, bonobo, chimp in the neuron state. These are all genes that are in the forebrain, that are not just in the forebrain, but make a forebrain neuron. The genes that are a little bit lower and higher in humans are the genes that make a hindbrain neuron. And the interesting thing here is that when I showed you the image for the NPC that looked like this, we saw that human, chimp, and bonobo, they all sort of looked pretty hindbrain, and the gorilla and the rhesus looked pretty forebrain. But now as we move to the NPC state, now that we move to the mature neuron state, we see this really stark differentiation where the humans are still lagging behind the other species, and the rest of the species are moving on to that more forebrain phenotype. One of the genes that is following this pattern is EGF, which in green, again, you can see the expression over time of the human EGF. In pink is the expression over time of bonobo. Red is pan-troglodyte's chimp. Purple is gorilla. Blue is rhesus. And you can see that the human is starting to express this gene EGF over time, which is actually a gene that's known to inhibit a forebrain phenotype and to drive a hindbrain phenotype. And so we're starting to figure out, just by gene expression, what genes are responsible for driving this neoteny, or potentially driving this neoteny in the human brain. Now, I've cherry-picked some genes here. I've picked genes that are known to be involved in neurodevelopment, but I'm not showing you the 20,000 genes that are expressed. One, because I know what these genes are doing, and because it's hard to show a lot of genes at once. But I want to know what's happening with all those other genes that we may not even have anything in the literature about, but may be important for hindbrain and forebrain patterning. And this is where that predictive model comes in, where we are predicting where in space our cells are coming from. So if we go back to that image, or we go back to that algorithm where we can predict where the cells are derived from, we can see that in the human, we have a more rhombencephalon, caudal, hindbrain phenotype. But as we move further towards chimp, gorilla, and rhesus, we start to get the forebrain phenotype. So it's not just these 5 or 6 or 20 genes that I pick and I look at from the literature, but overwhelmingly, the difference in expression is this separation across the forebrain and hindbrain. So this was intriguing to us, so we went in a little bit further and tried to pick out individual regions of the brain that may be driving this phenotype. We can see that there's an overrepresentation in humans of hindbrain structures, and in rhesus, there's an overrepresentation of the ventricular zone, that region that I told you earlier was responsible for high proliferation. Now, I've also been saying that the forebrain region of the genome, the forebrain region of the brain is developed later in time, and that the hindbrain phenotype means that there's some sort of neoteny occurring. But I haven't really shown that numerically, and I haven't proven that. So what I did here was I took the genes that are responsible for some of these regions, for example, the ventricular zone, and asked whether or not these genes really were predictive of being older or younger within a cell. So I used that second predictive model, and I estimated the age of the brain, or the cells, based on just these ventricular zone regions, which we know are being differentially expressed in the rhesus compared to the human. And lo and behold, yes, in fact, the humans are actually younger compared to the non-human primates, specifically the gorilla and the rhesus. So we're actually seeing, for the first time, molecular signatures of neoteny in the human brain based on gene expression. And also we're seeing it really early, way earlier than we would expect. It was even surprising to us that we picked up these signatures at the neural progenitor cell level, and we're picking them up through early differentiation as well. So I've told you that we have conserved dynamics of neurogenesis, and that also there's a regionalization that occurs as a function of evolution. And the last thing I'm going to tell you is this human-specific gene, or human-specific trends. So the last thing to ask is, what is actually inhibiting the humans from moving forward in developmental time? And in order to do that, I tried to identify genes that were expressed specifically in humans, and I found this gene, GATA3, that is increased in humans only, not expressed in the neural progenitor cell. And the interesting things about it is it's a primary pioneer transcription factor, meaning it can turn a cell into a different cell type. And this is important in T cell development, for example. If you knock this out, you inhibit T cell development. It is required for proper cell identity, and interestingly, it's required for differentiation of the hindbrain region of the brain. We've gone in and looked at where these GATA3 transcription factors are binding in the genome, and we found that they're actually binding to these ventricular zone genes, where they're binding to these regions of the genome that are progressing through differentiation. So we think that maybe GATA3 is actually binding to regions of the genome and inhibiting it from progressing and moving too quickly through time. So in summary, I showed you an examination of neurogenesis in multiple different species. That overall, this is a largely conserved process between species, but there are underlying differences that have evolved over evolutionary time, and that human neurons exhibit transcriptional neoteny. And with that, these are my acknowledgments. The head of our lab, Rusty Gage, Carol and Inigo for developing the systems, and the multiple people who have helped develop the tools and techniques that we're using along the way.